I think my job has become a little bit easier because of the crunch on time, because these are some really hard introductions, and so I get to be brief. But we're really excited to have Dr. Wilson and Dr. Marks here to talk about something that I think a lot of us are thinking about, which is what happens when we talk to the regulators about everything we've been discussing today. I don't think either of these guys needs an introduction, but briefly, Dr. Marks has, for the last several years, been the director of the Center for Biologics at the FDA. And Dr. Wilson has been a pioneer in the gene therapy field, starting a long time ago, and then maybe more recently, the isolation and discovery of AAV and substantial work in kind of evolving and discovering AAV and other capsids. And with that, I'll let you guys take over.
Great. Hey, Jim.
Great. You guys can hear me okay?
We can hear you. Can you hear us?
Fantastic. Yes. Thanks for asking me to participate in the meeting, and really appreciate the organizers for taking on a difficult topic that isn't adequately addressed in many other sessions. So appreciate that, and hope a lot will come from the meeting today and tomorrow. I'm glad to be here with you and Peter, but I thought it would be useful to begin with just for Peter to briefly talk about his background, because I think it's helpful to hear what his clinical training and his experience was as we begin to work with him as we move this along. Peter, I'll pass it over to you if you'd like to share with me.
Thanks. It's actually quite funny because I had flashbacks because I'm a hematologist oncologist by training. So I'm a medical scientist did my MD PhD at NYU, did internal medicine residency, hematology oncology fellowship here in Boston. Ended up doing a fair amount of support work for some of the transplant physicians, where there was a time when we were trying to do a fair amount of transplant into PRA positive individuals. Some of what you are using here in terms of various combinations were being used then, and we, including some of the first use of Rituxan in that setting. In the setting of trying to do immune modulation, so pretty familiar with some of this. And now over the past, after that, I've worked had a checkered past of working in industry and academics prior to coming to FDA about 12, 13 years ago, where I've been since. That's probably, I think, the interesting piece about this is that obviously now we're very interested in trying to facilitate advancing gene therapies. My area of the FDA handles vaccines, blood products, cell tissue gene therapy, xenotransplantation products, and clearly, gene therapy is something that over the past few years we've seen an increasing number of products approved, but also have encountered some of the limits, both in terms of manufacturing, in terms of exactly what we're talking about today, the issues that come up with AAV, capsids, and redosing, as well as starting to see over to the future where we might see more genome editing come into play. So that's where kind of we sit right now.
So Peter, you come to meetings like this, you're present, which we appreciate, and as rare diseases and gene therapies have been marginalized, FDA is leaning in, and as the opportunity that we all see may be passing us by, but based on the current situation, how are things working throughout your organization and sort of embracing a new day where we have to be innovative, not only in the technology, but innovative in the regulatory science? And because we will be, all of us work with you, but importantly your staff. So can you give us a little insight into how they're doing and how are they feeling about this?
Right. So the rare diseases, because in aggregate there are so many of them, and they are a great proving ground for getting gene therapy right, because we're not going to be making AAV for the masses right now, and we don't necessarily, right now the issue is that there are so many diseases potentially approachable by AAV, that it would be nice to deal with some of them, more of them than we are dealing with now. And so they're trying to lean in. And ideally, we'd get to deal with dozens and dozens and dozens on a yearly basis, but we're not there yet. And that's for a variety of reasons. Some within FDA's control, some outside of FDA's control, many outside of FDA's control. But one of the things we are trying to do is lean into the regulatory pathways to move things forward. And some of that has to do with, in the rare disease area, making sure that we are leveraging what we can do to get things through the development process. That includes trying to speed things through the process of development by having better communication. So one of the things we have ongoing right now is a communication pilot where we are changing. The goal is to potentially change how we communicate with people developing important products in the rare disease space, possibly in other disease spaces as well. Right now, for those of you who interact with FDA, you know that we have type A, B, C, D meetings. Takes anywhere from 30 to 75 days to schedule a meeting. Not the most efficient way when you're trying to get clarification of a manufacturing issue or a clinical design issue. One of the things we recognized during Operation Warp Speed for vaccines was that one of the ways you can really speed up development is you stop having scheduled meetings like that and you just have people ask questions as they come up. By asking a project manager who then interfaces with the team, and we get on the phone if necessary in real time. That actually took out months of development from the vaccines. Now the question is, can we do the same thing for rare diseases? So the hypothesis is it will make a big difference, but because it costs more money, it's going to actually cost resources. It takes more people to do that. We have to do a pilot to just demonstrate that it makes a difference. So we're doing that right now. We have four products in our center, 3 in the Center for Drugs, that are working with us on this kind of ongoing basis. And if indeed we're able to demonstrate by measuring, there are hard metrics, right? How long it takes to get from from your phase 1 to your phase 2 trial, how many times you end up on clinical holds, things like this that we're measuring. And if it does turn out, we will expand the pilot, the goal being to shorten that time of development. So that's one thing. The next piece is that as part of that, we're trying to make sure that we leverage the ability to do accelerated approvals as much as we can. The ability to use biomarkers in this area of rare diseases is critical. We can argue, we can go across the river and argue as much as we want in Boston about whether it's a good idea or not to do accelerated approvals, and that's a different debate. We can have that. Happy to have it for large indications. But in the rare disease area, I would put it to you that accelerated approval is critical to getting products to patients in need in a timely fashion. And it actually helps us abrogate the need for things like right to try, where people get stuff that's not safe potentially without any oversight. So I think it's a very good pathway forward for us. And it's a scientific one, right? If we understand the genes that are involved in something, we'd hopefully be able to get it right most of the time by looking at surrogate biomarkers or intermediate endpoints. So that's a second thing we're trying to do. And the third thing, although there are other things that we're doing in the manufacturing area, et cetera, but the third thing that I think is very important is that although you may not care about it that much in this room, one of the real problems for rare diseases is that when you get a product across the finish line in the United States, it doesn't mean you've necessarily gotten across the finish line outside of the United States. And having to do that rework if you want to introduce that product currently into the European Union or into other regulatory areas is costly and at times and takes time. That's actually a downer on development. Why? Because if you could more than double your market size without having much added effort, that would potentially be more impetus for people to go into and have profitable gene therapies. They would develop more things, but even more importantly, more people would actually get like products that help them. That's a novel idea, really novel idea, actually helping people. So we are working with the European Medicines Agency right now on a pilot where we are eliminating, we have, the reason why we're doing a pilot there is because we knew we had differences with EMA in terms of some of our manufacturing requirements. We also obviously have very different regulatory structures in terms of having, rapporteurs from different countries in the EMA and us having a more traditional single regulatory agency. So the goal here is to try to resolve as many differences as we can so that we can have simultaneous submission and potentially joint review of products, so that at the end of the day, simpler, if you could submit the same submission to both agencies and know that the same rules were going to apply, and if we can expand that further, that may also help in this rare disease space. I think it's a supply and demand issue, right? It'll help us. It'll help because we'll get the products to more patients more rapidly, but it'll also allow a greater revenue streams that will allow more money to hopefully come into the ecosystem. So there's kind of the three things we're trying to do, Jim. Sorry, maybe went on a little longer than
That's
you would
all right.
like, but to try to deal with this in the ecosystem. And then the final thing I'll just say is that our folks in the office are transitioning somewhat from an older model to a newer model of how we think about approvals. The older model was a frequentist model of statistics and a very strict kind of reading of, look, you have your statistical construct, you either meet the endpoints, you don't meet the endpoints, you do a certain number of trials, et cetera, to a more somewhat flexible totality of the evidence approach, where it's not that we don't care about statistics, it's that we're willing to use alternative statistical models, starting to look at Bayesian designs and other non-frequentist models to try to help products get across the finish line so that we can actually get and help more patients. What this probably means is that we may occasionally have products that will make it through to an accelerated approval that will have to be pulled from the market because they might not be able to be confirmed in a confirmatory trial. But hopefully that won't happen very often, and hopefully, probably some of the time when that happens, it's going to be not because the product doesn't work, but because it's sometimes hard to actually, in these rare diseases, figure out how to do a clinical trial that's able to demonstrate effectiveness. So I think that's something else that is something we're thinking about as we think about the whole accelerated approval process.
Great. So, Peter, it's something else that I've heard you talk about that may be worth bringing up here. Sort of relates to how can we best engage with FDA outside of the traditional meetings, but importantly, when we are involved in formal administrative processes with FDA, where there just is a difference of opinion. And, we're all trying to reconcile different points of view, patient advocacy groups that more and more are becoming more informed and more advocates. Then you have the sponsors, and then you have FDA. And as those discussions progress, and there does seem to be differences of opinion, which naturally we would have. How do you any advice in terms of how we would best deal with that?
Yeah, I think the first line of defense is good science and trying to reiterate the science that you've brought to it, and try to understand that, at least to just make sure that the disagreement is one that is based on a common ground of scientific evidence. Because you'd be amazed at how many times when we have disputes between sponsors or groups and the FDA, what we end up seeing is that they're not actually looking at the same data set. That's really, it seems like how can that be? But it sometimes happens. And so sometimes just ensuring that, okay, it's like marriage counseling. You want to make sure that each of the parties is actually hearing the other one before they decide to divorce. So you want to make sure that each of the parties actually, that that FDA has actually understands what you've brought to the table in terms of science. So that may mean asking FDA to mirror back to you, so what do you think we've brought to you? And if you hear something very different, well, it's probably a good idea to clarify it. Now, that's I think probably that's level one. The second level that sometimes occurs is we just have sometimes they're just fundamental disagreements that occur at a given level. And if you feel strongly enough that you're getting advice that doesn't make sense scientifically, or that is potentially arbitrary, then I think you can always ask for supervisory review. We have, what happens is oftentimes in a meeting you will have a group of reviewers and their chief, which is there's several levels down within our organization. Escalating to the next level up is something you can ask to do. And sometimes that supervisory review brings kind of a broader viewpoint to things. And it's a way to have discussion. And to be honest, if it's done politely, people actually don't mind it all that much. It's something that we've tried to help people understand. It's just a natural part of what we do. I mean, it happens to all of us. So yeah.
We, Peter, we went from a time when you had an avalanche of responsibilities in the COVID era, where cell and gene therapy took a back seat, and the resources you were able to commit weren't adequate, and timelines were delayed, and we didn't get meetings, and I think there was a lot of resentment, not to FDA, but rare diseases and a lot of others got the short end of the stick, understandably. But things have changed quite a bit. First of all, thanks to the work of you and your staff and many others, have COVID behind us. But where are we, with respect to your staffing, the numbers, the quality, the responsiveness? Assess where you think. I guess give yourself.
So Jim, thanks for those questions, because we actually just went over these numbers recently because of meetings as we're getting ready to ramp up for the next prescription drug user fee negotiations. What happened as part of the last prescription drug user fee negotiations was we were able to staff up with about 125 additional individuals through the center focusing on cell and gene therapy. 100 of those were in the actual office of that handles gene and cell therapy. There another 25 were sprinkled in the stats and communications and financial aspects of things that have to be taken care of. But also the inspections. I shouldn't leave those behind. So we have a greater staff. That has resulted in wait times that have gone down significantly for meetings. The number of it's also resulted in fewer clinical holds. The number of clinical holds has gone down by 50%. The number of meetings that have been converted to written response only now in the Office of Therapeutic Products has gone down to a very small number. And in fact, part of the issue now is that we're actually getting more requests by sponsors for written responses only than actually we're converting ourselves. So that's actually a good thing. And we'll try to keep improving on that. Hard to know how much more we're going to get in the way of personnel in this current climate. Don't know with the user fees, but I think we're in a much better place than we were. I also think that things we're doing in other parts of the center may actually help protect the gene therapy area. Without me going off topic too far, it was recognized that having to pull a lot of people off of their normal activities for pandemic response was very inefficient. It turns out that pandemic response, a lot of what gets done in that are very specialized things that if you do them routinely or more often, it can help expedite them greatly. We know that because the same teams that worked on COVID-19 worked on the Mpox response, and it was like night and day because the Mpox response was really, granted it wasn't the same magnitude, but it was handled so much more deftly. So we're actually putting together staff that will be handling that. So the hope is that if something does happen, untoward event like bird flu or something, we don't actually disrupt the rest of the ecosystem, because it really was a problem in the rare disease area that many programs really did pine away for a good year.
Peter, we're entering a new era that some of us who've been around for a while never would have expected, probably you. We're at a situation where most diseases don't have a gene therapy, a few have one, to an era where we're looking at second, third, fourth generation products. And there's some particular challenges here. And I think Duchenne's, for better, for worse, is going to be the example where this is, fortunately for the community good, you've had experiences, two reps, so to speak, Pfizer and Sarepta, but you've got another three that I'm aware of. We've heard about new capsids, and this is only the beginning, not the end of this revolution. But any thoughts about what have we learned from the first two, so we can start kind of refining this for iteration 3, 4, and 5?
Yeah, so I'm the first to say that I think that the Sarepta gene therapy is a generation one. It's got its warts, and there are believers and non-believers. At least I looking at the totality of the evidence and actually looking at it. Someone pointed out something to me today. Looking at it again today and looking back at our files, I'm still confident that we made the right decision there. But it's a generation one, and without the kind of more efficient transduction that we're talking about here. Probably it's a little naive to think that there's a 4-year-old has about 8 kilograms of muscle. And the idea that one dose of, and this goes back to something that Barry Byrne was saying, the idea that one dose of a gene therapy is going to be able to get into enough muscle, transduce enough muscle to deal with this, at least with that particular generation, was probably a little bit, it might have been a bit of a stretch. But the hope would be that as we come back with better capsids, the ability to potentially redose, it could be, exciting second generation products. I still don't have a complete explanation of why Pfizer got the results they did. It's not that I'm not willing to throw out microdystrophin entirely. I think what we will look at FDA is we'll look at each microdystrophin on its own. I think that means that if people bring us good data to connect microdystrophin to a clinical endpoint, their specific microdystrophin, we still have pathways forward that could lead to accelerated approval. It's just it has to be done on a case-by-case basis because we're going to take each microdystrophin on its own. I think there are things we still don't understand. So I think that's the way you get to second generation, third generation products, where hopefully we get to better and better products that are able to transduce more and more muscle, or have ways to be able to redose. And I think one of the issues that just to say that the conversation today, I'd be remiss not to say this. It's really exciting to look at all these strategies that are going along with trying to be able to redose. But just remember, those could add to the complexity cost of this therapy, and also then could get into the regulatory complexity of this, even the complexity with adverse events that get added on. So keeping it as simple as possible is probably an important thing as we're moving forward. Now, that means it may be that you can only keep it so simple, but that just means that those things will have to be taken into account when we're thinking about the regulatory pathways.
And one other question for you relates to the antibody as a companion diagnostic, or the antibody test. And our experience recently is we usually have pretty good success with FDA, but in Europe it's kind of getting messy. And there's something called the in vitro diagnostic regulation that is really, and where there's an in-house assay that is regulated differently than across countries. And what do you think the future is? Is there anything that we need to be doing differently, that you need to be doing differently, as we start to add these tests onto the assays?
Yeah,
treatments?
We're struggling a little bit with this. We've struggled with this a little bit in the United States, trying to make this simple, because you can't end up with a diagnostic test for 20 or 40 people that costs $20 million to develop. So that was something we're trying to address here in the U.S. I do think this is an area where it'll be kind of, once we get through our initial pilot with the EMA, this is one that we will probably have discussions with, but it's more complicated, as you just mentioned, because in some cases it's not actually, because EMA is not like FDA. They don't regulate devices in the same way. They do regulate combination products. It gets very complicated. I can't keep track of exactly all of the details, but we'll try to work through it with them. Yeah.
That would be very helpful, because it does create a lot of delays right now at a lot of different additional expense.
It's interesting, because once you get this is one of these things that drives me a little crazy. You get the regulators in a room, right, that are the people who are like mostly physicians, even attorneys who are familiar from different countries, and we all kind of agree, look, we gotta get rid of all of these differences if we want to have products that are only going to service 20 or 30 people a year in our countries to be able to get access to them. And everyone agrees there. And then you go outside the room and you go back, and there's suddenly like all sorts of bureaucratic stuff that comes in the way. So hopefully we'll be able to cut through some of that. It's actually, what I've been impressed with is actually has required a fair amount of high-level time to cut through some of the bureaucratic stuff, because it takes people at the level of agency heads to say, no, you don't have to do it this way. So hopefully we'll make some progress there. I do get the sense from Emer Cook, who's head of the EMA, that she's very interested in trying to move this forward. It sounds like the MHRA is as well. Health Canada is the same. And I know from talking to PDMA, they seem to be the same way. So hopefully we'll be able to get some tailwind here.
Very good. Well, I think Peter has a hard stop at 5 and maybe some travel plans. So I think we could probably conclude the session, and want to thank Peter for attending today, and again for partnering with us, and we're wishing you and all of us the best in the new year.
Yeah, no, thank you. And everyone, I just want to say thank you. It was really wonderful to listen to today's. Thank you, Jim. And it was really wonderful to listen to today's talk. I mean, just really impressive trying to solve I mean, the immune system is really a challenging thing, because it's just challenging. It's built to do things, and we're trying to fight what it's built to do. Sometimes it's really cool to see the different strategies evading or modulating. So hopefully, I think with a variety of different approaches, we'll see real progress here to be able to treat more patients. Because I do agree that when you have gene therapies for a small number of people, to have them be only available to an even smaller number of people is because of pre-existing immunity is even a bigger problem. So thank you for everything that all you do.
Thank you.
Thank you for the host.
All right, round of applause for Peter.
I think you have a few concluding remarks.
Is that me?
Yeah, that's you.
Okay, thanks. Well, it's a little hard to be very intelligent where I didn't participate in much of the meeting, but I do thank you for involving Peter in this discussion. And the FDA is going to have some challenging days ahead, and I hope we can be as supportive as we possibly can to work together with FDA. So and Peter and his team have done sort of extraordinary things to help in the rare disease space. I'd just like to end on one comment, as I've been involved in the field for a long period of time, and that was the problem of antibodies. And people always ask about the problem of antibodies without really having much of a solution. I actually viewed it as the dirty little secret of gene therapy was the antibody problems, or what people will say will be the immune problems. And it has not received the kind of support that it should, based on the impact that it is having and will have. They're complicated experiments. They're complicated experiments to model. But but my view is that some very important aspects of the impact of antibodies on in vivo gene therapy are not backed by a robust data set. For example, the threshold to determine when a patient is eligible or not, that's very much relevant in the context of the dose, the host, and the capsid. And I think we need to do better to identify what that threshold is. My sense is that some sponsors err on the side of defining a threshold to assure that everyone responds, but in the process, you may have patients that could have benefited and that aren't benefiting from that. And then secondly, what can we do, as we heard in the afternoon session, to try to render patients who were not eligible to be eligible using pharmacologic intervention. Peter was commented on that, and I think we do have to be mindful of the additional risks of certain forms of immune modulation. He said simpler may be better at this point in time, but I do think together PPMD and we should advocate for others that fund this research to try to get more data around threshold. How can you modulate one's immune system to render them receptive for gene therapy versus they otherwise would not. So and finally, where we're running into a lot of problems is when we try to conduct trials outside the United States, phase 1/2 studies. The regulations around the antibody tests are really becoming much more complicated in Europe, and I think with Peter's leadership, hopefully he can pull that together. It's in the entire time I've been involved in the field, I don't recall a meeting that was ever convened just to deal with the antibody problem. But congratulations to you all for pulling this together, and I really hope this is the beginning of a discussion that would continue and expand as we try to come up with solutions. So I'll pass it back to the organizers to conclude, I guess, this today.
So I'll be brief, but thank you again for coming. Thank you to all our speakers and moderators. We hope the discussion will continue. And I'll just take a couple more minutes to thank again the Ladders to Cures group here at the Broad and Parent Project Muscular Dystrophy, who really made all of this possible.